Hello, once again, my mortal audience, and welcome to this week's edition of Science Weekly. Today, we will cover the newest and greatest scientific discoveries over the past few weeks. Thanks for watching, and please enjoy. We might have found the key to unravel the mystery of the universe's expansion rate. A distant supernova, known as SNH0P, whose light has been magnified and tripled by the gravitational forces of the cosmos, could hold the key to unraveling the universe's expansion rate. SNH0P was discovered as a distorted smudge of light within data collected by the James Webb Space Telescope. This light originated from a galaxy whose radiance embarked on its journey over 10 billion years ago to reach us. Remarkably, this marks the second most distant supernova ever observed. What makes SNH0P even more captivating is that it belongs to the Type Ia category of supernovae, whose brightness serves as a vital metric for quantifying the universe's rate of expansion. However, it's not merely the supernova's distant nature that makes it intriguing. The gravitational effects on its light have introduced a time delay between different images of the supernova. This phenomenon presents an exciting opportunity for scientists to address one of cosmology's most profound enigmas, the Hubble constant, denoted as H0, representing the universe's expansion rate. An international research team, led by cosmologist Brenda Fry from the University of Arizona, has submitted a paper outlining this discovery to the Astrophysical Journal, which is currently accessible on the preprint server archive. In their paper, the team writes, "We present herein an overview of the initial science results in the combined JWST pearls and DDT imaging and spectroscopic observations in the G165 cluster field, and of the discovery of the SN, which is dubbed SNH0P, for its potential to measure the time delays between the images, and from it, a value for Hubble's constant." This study is the first in a series of papers whose objective is to investigate SNH0P, the cluster, and the lens sources. The Hubble constant has remained a persistent challenge in cosmology. While we understand that the universe is expanding at an accelerating pace, determining the precise rate has proven elusive. Two primary methods are employed to calculate this rate. The first, referred to as standard rulers, typically yields an expansion rate of around 67 kilometers per second per megaparsec. The second method, known as standard candles, results in an estimate of approximately 73 kilometers per second per megaparsec. Standard rulers encompass phenomena like the cosmic microwave background, a relic of the Big Bang, and baryon acoustic oscillations, which are frozen-in-time acoustic waves. Standard candles rely on objects with known intrinsic brightness, such as Type Ia supernovae or Cepheid variable stars. Knowing an object's intrinsic brightness enables the determination of its distance. Type Ia supernovae, in particular, possess a consistent intrinsic luminosity. However, beyond a certain distance, observing individual stars or even supernovae becomes exceedingly challenging due to their diminutive size. This limitation renders standard candles less effective for gauging the expansion rate of the distant universe, with a notable exception: gravitational lenses. Gravitational lenses occur when a sufficiently massive object bends space-time significantly, creating a curvature. This curvature causes light passing through it to follow a curved path, resulting in fascinating effects observable from our perspective, including magnification, distortion, and replication of a single light source. Brenda Fry and her colleagues stumbled upon SNH0P during the James Webb Space Telescope's extensive observations of the universe, which unveiled numerous intriguing gravitationally lensed objects. In multiple observation sessions, data revealed a galaxy called ARC-2, with its light being lensed by a massive galaxy cluster in the foreground. Analysis of these observations disclosed three distinct dots of light, identified as the radiance of a Type Ia supernova. In conclusion, the researchers anticipate that the time delay estimates, derived from various lens models, photometric data, spectroscopy, and a weighted combination of these estimates, will eventually yield a value for H0. The revelation of this value, and its alignment with other measurements of the Hubble constant, promises to be profoundly exciting for the field of cosmology. Really interesting stuff. Anyways, our next story is more of a follow-up on older news. Off the shores of South Africa, a fascinating natural drama has been unfolding involving orcas and great white sharks. However, recent times have seen a mysterious disappearance of great white sharks from these waters. Scientists propose that the surviving great whites have shifted eastward to avoid confrontations with the orcas. For a significant period, South Africa's False Bay and Gansby shores were the scenes of a perplexing phenomenon. Deceased great white sharks were turning up, notably missing a crucial organ, their livers. In a chilling display of precision, orcas had been extracting the livers from these sharks, resulting in their demise. Then, a change occurred. The great white sharks ceased washing ashore. They refrained from swimming near these shores, which were some of their most renowned habitats in South Africa. It was as if they had vanished. The decline of white sharks was so abrupt, so swift, and so unprecedented that numerous theories started circulating, remarked Michelle Jewell, an ecologist at Michigan State University Museum, in an interview with Hawkeye magazine. Concerns arose that the overfishing of both the shark's prey and the sharks themselves might have substantially reduced their population. However, a recent study published in the October issue of the journal Ecological Indicators presented a different perspective. The great whites hadn't perished. They had merely relocated to safer territories. By examining records of interactions between humans and sharks, scientists deduced that the South African shark populations had migrated eastward, finding refuge in areas like Algoa Bay and the KwaZulu-Natal coastline. We know that predators exert significant influence on the movement and habitat choices of their prey, so this isn't entirely unexpected, Jewell conveyed to Hawkeye magazine. The study employed a process of elimination to investigate various potential explanations, including a decline in prey availability in the region where the sharks had disappeared, and the rapid reproduction of sharks in areas where numerous new sightings were reported. Ultimately, the scientists concluded that the most plausible explanation was the great white's flight to evade ongoing encounters with orcas. Folks, you won't believe this next one. You just can't make this up. Mathematics has a peculiar way of revealing patterns in the natural world, and a curious connection between the formation of zebra stripes and the swimming behavior of sperm is a striking example of this phenomenon. Drawing inspiration from how stripes and spots are believed to develop in a wide range of organisms, from zebras to giant pufferfish, researchers delved into the intricate patterns created by the whirling flagellum of sperm. This research harkens back to the work of Alan Turing, the renowned English mathematician who, among his many accomplishments, proposed a theory for the emergence of wavy or spotted patterns in the natural world. Turing introduced the concept of what we now call reaction diffusion systems, suggesting that biological patterns could arise from the interaction of two molecules diffusing through space. In 1952, Turing laid the groundwork for chemical patterns through reaction diffusion, explains Hermes Bloomfield, Gadea. We demonstrate that in the microscopic world, the flagellum, which serves as the building block of motion, employs Turing's concept to create patterns of motion, propelling sperm forward. Before delving deeper into this, it's essential to recognize that despite Turing's brilliance, reaction diffusion models have long been a subject of debate, with experimental biologists questioning their realism. Like any mathematical model, they simplify the intricate processes of nature into a few key parameters. That being said, mathematical models can be invaluable for conceptualizing complex behaviors, such as the swaying motion of a sperm's tail or flagellum. By breaking down the fundamental components of flagellar motion into a simplified model, we have found that reaction diffusion dynamics can effectively account for the observed patterns in flagellar beating. Instead of chemical substances diffusing and reacting freely in space, what Cass and Bloomfield-Gadea describe is akin to a tug-of-war reaction kinetics involving molecular motors anchored within the flagellum structure. These motors create shear deformation, which diffuses along the slender, flexible appendages. In simpler terms, the flagellum's rhythmic motion originates from molecular motors at its base, generating oscillations that propagate along its supple structure in wave-like patterns. This process can be modeled using reaction diffusion dynamics, resulting in the formation of band-like patterns, as depicted in the accompanying image. Recognizing the limitations of their model, the researchers went a step further by comparing their simulations with experimental data from previously published studies involving sperm and Chlamydomonas reinhardtii, a single-celled green algae. Recent research has unveiled the remarkable complexity of sperm mechanics, characterizing the axoneme, the molecular machinery that propels flagella and sperm, as an intricate assembly comprising hundreds of distinct proteins. Yet Cass and Bloomfield-Gadea found that their simplified model of flagellar mechanics accurately captured flagella's movement patterns, closely mirroring those of eukaryotic flagella. We demonstrate that this mathematical recipe is shared by two very distant species, bullsperm and Chlamydomonas, a green algae widely used as a model organism in scientific research, indicating that nature employs similar solutions, notes Bloomfield-Gadea. Although these models may initially seem like intellectual pursuits, simulating the workings of sperm flagella carries significant implications for our comprehension of sperm motility, a factor with direct relevance to male fertility. Nature never ceases to amaze with its weirdness. Now for our last story. The presence of ice on the moon is well established, but the origin of this lunar water has been a subject of ongoing investigation. A recent study suggests that streams of electrons, indirectly originating from both Earth and the Sun, may be contributing to the formation of frozen water on the lunar surface. These electrons impact the moon as it moves in and out of Earth's magnetotail, a region of space that our planet leaves behind as it travels through the cosmos. Within the magnetotail lies a plasma sheet composed of highly charged electrons and ions drawn from Earth's atmosphere and the solar wind emitted by the Sun. Previous research has explored the role of the magnetotail and the broader magnetosphere in the creation of lunar water. The magnetosphere arises when Earth's protective magnetic field deflects the solar wind from the Sun, resulting in various effects in its wake. This provides a natural laboratory for studying the formation processes of lunar surface water, explains planetary scientist Shuai Lee from the University of Hawaii at Manoa. When the moon is positioned outside of the magnetotail, the lunar surface is bombarded by solar wind. Inside the magnetotail, there is almost no influx of solar wind protons, and it was expected that water formation would decrease significantly. Nevertheless, remote analysis indicates that this isn't the case. Earlier research suggested that hydrogen ions from solar winds were responsible for generating lunar water. However, it appears that water formation continues even when the lunar surface is shielded from solar winds within the magnetotail. The researchers propose that other factors are at play, particularly electrons. One possible mechanism is that high-energy electrons interact with the lunar soil, releasing trapped hydrogen, which can then combine to form water. In the magnetotail, there may be additional water formation processes or new water sources that are not directly linked to the implantation of solar wind protons, Lee explains. Specifically, the radiation from high-energy electrons produces similar effects to solar wind protons. To confirm this hypothesis, further observations and experiments will be necessary on the lunar surface. Nonetheless, this proposal is intriguing and adds to the many avenues of scientific inquiry aimed at understanding the origins of water on the moon. And that concludes our report for today, folks. Join us next week, when we will cover more news in a similar manner. If you would like to support us, please don't forget to like and subscribe. Until next time, mortals.